We have arrived at our last module for the class, Module 5. And for this one, we're going to cover the central processes and the essential processes of the central dogma, which starts with DNA replication. So for this part, we have the following learning objectives to describe the steps of DNA synthesis, to compare it in prokaryotes and eukaryotes, and then to explain what could happen in terms of the cell cycle and the effect of mutations. The central dogma describes the flow of information from DNA to RNA to proteins. As a reminder, this first process involves the DNA replicating itself. The cell cycle includes both interphase and mitosis, and the DNA must replicate itself before the cell can divide. Now this occurs in S phase. So shown here is interphase and mitosis being a small part of interphase, and S phase for DNA synthesis is a part of interphase. And at the end of DNA synthesis, we're going to end up with chromosomes that have 2 chromatids. DNA replication is a semi-conservative process. And what this means is that the 2 new helices of DNA will each have 1 parent strand, 1 old strand, and 1 daughter strand, which is new. This, keep in mind, we have the sequence of DNA that we need to remember and be aware of. The strands have what we call complementary base pairing. So anytime you see an A, on the other strand, you're going to see a T, and with a G and a C. And remember that DNA has a direction to it. So it's made in that 5' to 3' direction, and the strands are anti-parallel to each other, which means they run in opposite directions, 5 to 3, 3 to 5. So the DNA synthesis involves both the initiation, so we want to answer the question, how does replication start? A synthesis step, which we want to answer the question, how does replication proceed? And then proofreading, so how are errors discovered? DNA replication begins at a replication fork, and in bacteria, the DNA proteins are going to bind to a consensus region and then use this consensus sequence. The DNA proteins will bind, and then they're going to use helicase, which is going to unwind that DNA helix, and that will be stabilized by our single-stranded binding proteins. In eukaryotes, the initiation is a little bit more complicated and involves multiple origins of replication with a complex of proteins that will help with initiation and will also open and hold open the DNA. So again, helicase is going to do that unwinding. Single-stranded DNA binding proteins are going to hold it open. And here we also use a topoisomerase, which helps prevent supercoiling as the DNA helix twists in the other direction to open up. Because of the directionality of synthesis, each strand of DNA must be synthesized differently. So here's our replication fork. And you can see that for one side, it's very easy to make that 5' to 3' heading into the direction that the DNA is opened, so towards the replication fork. But because of the anti-parallel nature of our DNA helix, the other side, we call it the lagging side, if it makes it in the 5' to 3' direction, it's moving away from the replication fork. And so we'll need to make a series of fragments as the replication fork moves and as the DNA helix opens. And we call these fragments Okazaki fragments. Now these fragments then need to be put together, and this enzyme called ligase is the one that puts these fragments back together. How does the DNA strand grow? Well, it grows through an enzyme called DNA polymerase. And DNA polymerase is going to add nucleotides to an existing chain. It cannot do what we call de novo synthesis. And so it has to start with something. And that something is a primer. And the primer here is made by RNA primase. And so it's made of RNA nucleotides. And then the DNA polymerase can bind and grow the chain. Now here, DNA polymerase 3 is the one that makes the chain, and then DNA polymerase 1 will come back in and will replace that RNA primer with DNA. And again, ligase will come and seal all the gaps. Ligase seals everything together. Okay, so in this video, we cover the process of transcription, and in the next video, we're going to then talk about, or in this one we talked about replication rather, and in the next one, we're going to talk about transcription.